morning, everybody. Welcome to Bioconductor 2021 and this workshop session on multi-omic integration in cBioPortal and TCGA data with multi-assay experiment. We're going to start the talk shortly, but first a few announcements. If you have questions for the speaker, please enter them into the Q&A tab. You'll find the Q&A tab on the right under the conversations. If you have questions for the speaker, so put them there. And if you also will notice that while you're putting questions in, you may notice that other people have put in the same question. So if you see the same question, be sure to upvote the questions that exist rather than putting the same questions into the tab. So make use of that feature to keep things from being cluttered. And if you need to ask a question live, please feel free to use the raise hands feature and the moderator can bring you on stage. There are also a variety of fun emoticons you can click on down below to give feedback to the speaker for applause or to tell, and to indicate that you found the thumbs up button. Great. If for some reason you need to leave early, a video of the session will be available a couple of hours after the session is completed. And now let me introduce to you our speaker, Marcel Ramos.
Thanks, Mark. Welcome, everyone. Thank you for coming. I'll be giving you some updates on multi-assay experiment, as well as introducing you to our, I guess, suite of packages now. So let's begin with starting up our instance, and I will share my screen here. So in order to easily find our slide deck here, I created this tiny URL. So if you go to tinyurl.com/multi-assayExperiment, that will provide you the relevant links for the workshop here. And one of them is our package down website, which you can consult. We will consult for all of the vignettes associated with the workshop, and the workshop instance website, which is the app.orchestra.cancerdataSite.org location. So once again, if you missed that, go to tinyurl.com/multi-assay-workshop. You'll get access to these links. Our workshop instance is listed as multi-omic integration of cBioPortal and TCGA data with multi-assay experiment. And if you want the direct link, it's this workshop ID 73 down below here. So I'll do that myself to launch the instance. If you're at the page, if you logged in, it should be on page 5 at the top. So click launch on that, and we'll wait a little bit for that to cook. And what we'll do is I'll begin with the introduction to the workshop. So the purpose of the workshop is to introduce you to multi-assay experiment and the associated packages with that, including curated TCGAData, cBioPortal, and TCGAUtils. So I'll give you an overview of what the packages do, and then we'll try building a multi-assay experiment data structure from scratch, and we'll look at cBioPortal. We also have a tutorial for that, and we will, if we have enough time, we'll go over some analysis examples using multi-assay experiment. So here at this web page that I'm looking at is that first link in the slides, which is our package down website. So that's where we're starting. And if you click on the top button here that says workshop, that will take you to the main workshop contents. So the password is RStudio, username RStudio. I think this was a little bit out of date here. And I'll give you an overview of the packages. So multi-assay experiment. Well, let's start with a poll and see how many people have used multi-assay experiment in the past, and maybe that'll be a good starter here. So you should be able to see a poll on our platform. So I see the numbers ticking. Around 80% say no, which is good. This is meant to introduce users and everyone to multi-assay experiment. And about 20% of you have answered yes. So that looks good. So we'll start from the beginning here, and I'll guide you through all of these packages. So the first one is multi-assay experiment, which is the infrastructure package for coordinating multi-omics experiments. So if you're running several experiments and you need a way to handle all of these experiments along with some phenotype data, multi-assay experiment is the package to use. We also have curated TCGAData, which takes multi-assay experiment, the infrastructure, and provides TCGAData through Experiment Hub, so that you can download from about 33 different cancer types and get a nice multi-assay experiment from a single bit of our code, single line of our code. cBioPortal is the interface to the cBiogenomics portal that started at MSKCC, and they're collaborating with other institutions as well. They have about over 300 study data sets that can be more or less imported as a multi-assay experiment. And I'll make this bigger since I think that should be better. So that's cBioPortal. It gives you programmatic access to the data sets on the cBiogenomics portal. TCGAUtils is more of a complementary package that allows you to do some operations with TCGA data that simplify the workflow. And more recently, we've worked on single cell multimodal, which provides single cell data from various modalities and technologies. So to start off here, I will go over the multi-assay experiment infrastructure, and we have a graphic to represent it. On the bottom left, you can see that we have several experiments where they're not necessarily of the same shape, so they can have different dimensions. And on the right-hand side, we have a sample map which coordinates these experiments with the call data at the top. And the call data has the patient information for all of the patients in the data. So those three pieces of information work together to provide a coordinated representation of the data. And you can have multiple samples per patient within an experiment. You can see as denoted by these green lines here on the left. So let's check on our workshop and see if it's done. So it looks like for me it's ready. Please don't look at my link. Otherwise you'll boot me off. But here I'm going to log in as RStudio with the username RStudio and the password RStudio. And don't say the password. So this is our platform here. I'm going to bear with me while I change some settings here so that it's easier to see. Here, that should be good. Okay, so this is our instance for the workshop, and if you click on this description file, you can see that we've created a package for the workshop with dependencies, all the dependencies required for running the workshop. So the first thing that we can do is to do our trusty help function and do help packages equals multi-assay workshop. Or package singular. So once you do help package equals multi-assay workshop, you will get this pop-up here where we can click on our vignettes section, and we can have a look at all of the vignettes that come with our workshop. So the first one is the integrating multi-omics data with multi-assay experiment. So you can see that we have a runnable version of what we were just looking at on the website here. So one note here is that if you want to come back to this workshop, you can use our Docker version of the workshop, which is listed as this mr/multi-assay workshop : latest, and you can run that if you're using Docker to spin up this instance on your own computer at a later date. Although the orchestra images are available until the end of the conference, more or less, as far as I know, and you'll be able to access it during the conference. So here we have our vignette that we were just looking at on the website. So we have everything in one place now. And we also have a tutorial here that we can run. So if you go back to your files, go to Inst, Tutorials, click on Multi-assay Experiment, and you should find an RMD file with the name exercise_multi-assay. So that is a Shiny-based Learn R tutorial that we can click on the Run document here. And that will spin it up for us. And we'll get a pop-up. If your browser does not have pop-ups enabled, you can make an exception here to see that pop-up. See, I got the notification here. On the top right, I can click on Always Allow Pop-ups from this website, then click Done, and then try again. And then I'll see the little window show up here. Okay, so this is our tutorial for building a multi-assay experiment structure from scratch. How many people have been able to get this? Click on the thumbs up. All right, looks good. Unless a few people are spamming, then you can't really tell. Okay, so here we can go through this tutorial to see what components are required to create a multi-assay experiment from the constructor function, which has the same name. It goes through our schematic here. Click Continue. So we do have a small multi-assay experiment data set included in the package with the name mini-ACC. So this is adrenocortical carcinoma. So it's taken from real data. It's a subset of the actual data set. And the instructions here say use the experiments function to take the experiment data out of mini-ACC. So what we're going to do is I'm not sure if this is big enough for everyone to see. But I hope that's better. Okay, Okay, so we're going to use the experiment function on this object, and then we're going to run the code, and that will give us the experiment from the many ACC data sets. So you can see that multi-assay experiment supports several different omics types, mutations, microRNAs, protein data sets. So, and you can notice that the columns here are not necessarily uniform across all of the experiments as we saw in the schematic. So that experiments function will allow us to extract only the experiment list from the multi-assay experiment representation. All right, so now we can look for the phenotypic information from the object so that we'll use this callData function to extract the phenotype information. So if we callData on mini-ACC and run that, and you can see that it's a data frame with 92 rows that correspond to the number of patients in the data set, and 30 columns, which are the variables in the data. And you can see some of the data like years to birth, vital status. So you see some of the variables that are included in the example data set, mini ACC example. Let's click continue. So now we're going to extract a column from the callData here. There are a couple of ways of doing that. You can do callData first and then extract a column. Do callData and then $race, for example. Or we provided a more convenient function to just use $race, which will go directly and extract that variable from the multi-assay experiment. So either one should work here. So you can see we have the same result in both outputs here. So that's working with the callData. Now we're looking at the assays. So in your workflow, you might want to take all of the data and send it off to another software for analysis. So you could use this assays plural function to extract a list of matrices for analysis. So that will give you a list, and then you can use that in other packages to work with if that's the input, unless the package supports multi-assay experiment, then you can simply pass along the multi-assay experiment. But assays will give you that list of experiments as matrices. So our third representation here is the sample map, which coordinates all of the experiments and the phenotype information. So the extractor function that we can use to have a look at the sample map is sample map, lowercase s, capital M, and that will give you a graph representation of the assays, the patient IDs, and the sample IDs in the data. So this is how the coordination happens, and when you take a slice of the data, this is what gets updated as you perform subsetting operations or remove assays from the data. So that's our sample map, and now we'll look at the we have a little quiz here. So let's test our knowledge here. What function do we use to extract the experiments from mini-ACC? So there's experiment list. So it's lowercase e experiments. submit that. That looks good. So this is a common mistake. Our experiment list structure is a class, but the way to extract that data is with the lowercase experiments function on a multi-assay experiment. So yeah, that would not work here. Okay, so now let's look at the constructor function. The constructor function requires 3 main arguments that all correspond to the components of the multi-assay experiment. So that's the experiments, the call data, and the sample map. So you don't always have to have a sample map. You can simply provide a set of experiments and call data if the column names and the row names in the experiments and call data are the same. So there are some conveniences that we provide to reduce the amount of data munging that you have to do. So and we also provide a function to help you build your multi-assay experiment called prep multi-assay, and that will point out any things that need to be resolved in order to create a multi-assay experiment. But because we already have an example multi-assay experiment, we're going to use that. So we'll practice extracting all the components. So if we do lowercase experiments, we'll get the experiment list from the mini-ACC dataset. So that could be our input. So let's start this constructor function. So I'll do multi-assay experiment, and then I'll do experiments equals, add some space here. That's our first input, experiments, and then call data is the second input, and that is call data mini-ACC. So here we're extracting the elements and rebuilding the multi-assay experiment with the constructor function. So that's why it looks a little interesting here. And lastly, the sample map. So you can leave that out if you'd like, as long as your call data and your experiment names, or your call names in the experiments match. So that's what our constructor function would look like. You have the experiments, call data, and the sample map, and then you can run that code. And that will produce the original mini-ACC. So that's how we create our multi-assay experiment, and that was our tutorial here. So now let's look at are there any questions? Why have experiments versus assays? What is the real difference there? So experiments will return the actual classes that were input at the beginning. So and assays will reduce all of your data to matrix format. So multi-assay experiment supports several classes that have a rectangular shape. So you can include single cell experiments, summarized experiments, or ragged experiments within a multi-assay experiment. When you run that experiments function, you'll get those classes back. But when you use assays plural, you'll get more of a reduced format as a list of matrices. Okay, so now let's look at our second package in the list here. Sorry, I'm switching back and forth. Our second package is cBioPortalData. So it takes about 300 studies and gives you a multi-assay experiment representation. So we can have a look at the tutorial to see how to use the package. We have two main functions in the package. One is to download the entirety of the data, which is our cBioData pack. And the other function is to make use of the revamped API with caching so that you can request sections of the data that are hosted at cBioPortal. So if we go back into our instance here, our studio instance, sorry. If we go back into our tutorials folder, so if you go home, then go to inst tutorials, you'll have a cBioPortalData RMD. So we can run this as well. So make sure you have those pop-ups disabled or that option disabled. And we're running this tutorial here to get familiar with what cBioPortalData does. So here we'll learn how to use cBioData pack and cBioPortalData. So the cBioGenomics portal also provides TCGA data, and along with other study data sets. So there's a little bit of an overlap, but we provide a programmatic interface to their data, so that's pretty convenient. So we can see a list of data sets by doing a get studies. So first, I think we need to, these instructions are not right here. But let's do this instead. So what we're going to do is create an instance of the API. So what we have to do first is use the cBioPortal function to read the API and create an instance of it. And then we can do get studies cBio. So let's run that code to get a list of what's available. So you can see that we get a list as a table here of all of the studies in the cBioPortal repository. You get a description, public study, any associated PMIDs, and some more metadata. So I think what's more important for us when using the software is that we want to know what the study ID is. So you can see that here we have a list of the study IDs that we will use to query their service. So that is how we learn what studies are available in cBioPortal. So you can see that there are about 316 rows here, so that's quite a number of studies. All right. So I think this has changed recently, so I will skip this. Okay, so now we'll try using our cBioData pack function on the ALL leukemia dataset from St. Jude. So we'll do cBioData pack and then run that code. So you can see that this is a small study data set, so it has a couple of files in it related to mutations. And when you run that function, you get a multi-assay experiment back with a ragged experiment representation, two ragged experiment representations in that multi-assay experiment. And we'll go over what ragged experiment does shortly. So that's the basics of using cBioData Pack. You just need to input your study ID, and we do have a list of, if you go back to that get studies function, it will tell you whether or not, because there are some issues with data sets and in terms of data cleaning and the IDs, we can't build all of the data sets currently as multi-assay experiments, but we have more than 80% building as multi-assay experiment. So if you have a very important data set that you want to look into, and we can help you look into it and make it work with our the functionality that we provide. So you can open up an issue on our GitHub page. Should be listed in our description file, so under the bug reports field. So let's look at that. I'll do package description and do portal data. It's rerunning the cBioData Pack function, but it's okay. So here, when you do package description, you'll get the relevant link here. So there's bug reports. That's where you can file an issue if there's a data set that's not working, and we'll be happy to work with you and the rest of the cBioPortal team to fix any issues with the data. All right. So that is the prepackaged data that comes in the tarball. The other version of the data is the API, which allows you to take slices of the data, and that requires here what we did earlier, a cBioPortal instance. And we'll do this again just to reiterate here. So you do cBioPortal, pull up that instance, and we can have a look at what the contents of it are. So it's a representation in R where it will show you all the endpoints and a summary of these endpoints in cBioPortal. So you'll need this cBio or this instance for most of your queries here. So that's what you'll have to include in the main function, the cBioPortalData function. Okay. Okay, so for submitting the cBioPortalData function call, we need to include a study ID from our get studies. This is not correct. If you're on the release in Bioconductor release version, studies table should still be available, but we're changing this for the develop version and having everyone use the get studies API endpoint, which is more reliable than the studies table. Studies table doesn't get updated as often as the actual API. So we're removing that from the package and pointing users or everyone to the get studies function. So that's what we need. One component is the study ID. The other one is a set of genes that you're interested in, or a gene panel ID. And a by argument, which will indicate whether you want Hugo symbols or entree gene IDs, and then molecular profile IDs. So in our examples, we can have a look at what gene panels returns here. So this function will query the service and give you a list of all the gene panels that are available by ID here. So you can see these are study gene panels. You can use those or the genes that you'd like to have queried. The molecular profile IDs can be obtained with this molecular profiles function and the API object, which is cBio in our case, study ID, which in this case we're looking at ACC_TCGA, and we'll run that to see to populate a table of all the molecular profiles available for that study. So you can see here we have the alteration type with methylation, copy number, mutations, and what we're interested in is the ID, the molecular profile ID. So that's what we want to use when we're generating that query via the cBioPortalData function. Okay, so I think we'll use one of these. We'll try it maybe with RPPA here. All right, so we'll bring it together and we'll say we want the ACC TCGA study ID with this impact 341. This is an example. You can again use your set of genes as well, and a couple of molecular profile IDs, RPPA, and mutations. So I'll copy those over. So that goes under the molecular profile IDs input. And then we want this by entree gene ID. So as you can see, it will query the service and then give you those two assays for all of the patients, because you can also specify some patients, but by default it will give you all of the data available for all the patients in that molecular profile ID. So we have a summarized experiment and a range summarized experiment to work with now after querying the service. All right, so now we have some trivia. What's the difference? So this will help us understand the difference between the two functions here. So what function provides prepackaged cBioPortal study data in its entirety? Data, load, cBioPortal, or cBioData pack? As you may have guessed, cBioData pack here. What function can you use to obtain the cBioPortal data as served by the API service? So it's the same name as the package, cBioPortalData. And what components you need to create a cBioPortal data function call? You need the study ID, the genes or gene panel ID, molecular profile ID. So it looks like all of the above. Great, so now we have some idea of what cBioPortalData does and how it works. And now we can go through the following packages here. So we'll look at curated TCGA data. So this was part of a pipeline that we worked on to take data from the Broad GDAC firehose pipeline and serve it as multi-assay experiments. So it makes use of a number of packages like our TCGA toolbox and TCGAUtils to serve these data sets for you. There are other tools that allow access to TCGA, like Genomic Data Commons, TCGA BioLinks. But we provide the data as a multi-assay experiment, so it's a little bit easier to use and manipulate. So we have some vignettes for seeing what studies are available there. And if you click on that reference vignettes links, you'll be able to see those. And we have a description of what omics types are listed there in TCGA data, curated TCGA data. And more recently, we have single-cell multimodal, which provides data sets in different representations, including out-of-memory data sets using the HDF5 format and the matrix market format. And we provide data from several technologies like SCNMT, 10x Multiome, SeqFish, ExciteSeq, Scope2, and others. And thanks to our collaborators like Dario Vigelli and Kelly Eckenrode and others that help make this package possible here at single-cell multimodal. So feel free to check that out if you are analyzing single-cell data. And last but not least, we have TCGAUtils, which gives you a host of utility functions for separating sample types, because the data that you get from curated TCGA data includes tumor as well as normals sometimes in the data. So you want to separate those out, and we provide a function to do that. And this is a schematic of what our pipeline looks like, more or less. So we make use of multi-assay experiment as the central data representation for all of these packages. So hopefully you are familiar with the SummarizedExperiment class. Multi-assay experiment is modeled after this class. And if you're not, here we have, so the difference between SummarizedExperiment and multi-assay experiment is that SummarizedExperiment has these rectangular assays that can't vary in dimension. So you have a set of genes that are consistent across all of these assays and a set of samples in a SummarizedExperiment. For multi-assay experiment, you can have this whole summarized experiment as well as a single cell experiment within the multi-assay experiment and still be able to coordinate the phenotype data and all of the experiments within the object. So, and there's a range SummarizedExperiment derivative, which allows for ranges in the rows. So you can do some queries with the genomic ranges functionality and subset using that, which multi-assay experiment also supports. So if you have the G ranges and a set of experiments that all have, say, for example, a range SummarizedExperiment, you can subset your experiments with that G ranges. We've seen RaggedExperiment come up a few times in the examples. This class we worked on because we needed something to represent mainly mutation data from a set of patients that don't have the same measurements across. So we developed RaggedExperiment because it allows every patient to have a different number of measurements for each patient. So we can take a G-ranges list as input and convert to RaggedExperiment as a data structure that will conform to the multi-assay experiment requirements. And the multi-assay experiment requirements are minimal. The requirements are that the data structure is rectangular. So if you can do like a bracket subsetting and have dimensions for rows and columns, then it will fit in a multi-assay experiment. So it can be as simple as a matrix or as complicated as a single cell experiment. So they're all supported. So RaggedExperiment is one of those that takes the G-ranges list and represents that G-ranges list as a rectangular representation. So this is our schematic, and there are a few things that you can do with RaggedExperiment. You can take simple representation and create a sparse assay from all of the samples in the RaggedExperiment, or you can combine those ranges in a way that does it a bit more intelligently here. Like in QReduce assay, if you have a set of G ranges that you're interested in, you can take that G ranges and restrict all of your ranges in your RaggedExperiment to that G ranges. So check out the vignette for more info, and feel free to ask questions on Slack if you have questions, or on the support site as well. Okay, so let's go over a derivative of multi-assay experiment, which is our matched assay experiment. And what that does, it ensures that all of the columns in all of your experiments are uniform. So although multi-assay experiment supports those experiments that have unequal columns throughout, you can coerce to matched assay experiments so that they're all of the same length. So if you do something like as multi-assay experiment and then matched assay experiment, that will do a set of operations that will make that data uniform and have the same number of columns throughout all the experiment. Okay, so I think we should have a look at our a demo for multi-assay experiment and curated TCGA data. So let's go back to our RStudio session. And what we're going to do is sort of remove some of these out of the way. So here we're going to load curated TCGA data. So the neat thing about this, the RStudio interface here, is that you can highlight something and then click Control Enter, and it will send it to your console pretty conveniently. So here I'm doing library curated TCGA data to load the package. And we'll do something like ACC. We'll load the ACC data set from curated TCGAData. So we'll do ACC gets curated TCGAData. ACC is our disease code. Add those. Maybe this is better in a script. So disease code equals ACC. What assays do we want? Let's try number, alterations, patient. Let's try those and see what we can get. We can also do a query and see what we have available. So if you just simply do ACC asterisk and then try run equals true, that will give you a small table. Oh, and I forgot to include my version parameter here. So if you so this will give me a nice table here of all of the resources that are available for this this disease code, and it will ask you if you want to create a directory for caching. I'll say yes, and then it will go into Experiment Hub, query what files are available under that that disease code, and we'll get a table that looks like this. So we have SNP data, gistic, methylation, mutation available for the ACC disease code. So we can do RNA, mutations, and maybe CNA. So let's run that and include a version here. So we want version 2.01, which is the more recent one. And we want dry run equals false so that we can download the actual data. So that will go into Experiment Hub and pull those data sets that match those these fuzzy matches here, and put those in the multi-assay experiment. Okay, so now that we have it, we can do ACC in the console and we'll see our contents. And you can also do call data ACC. Look at call data, there's a lot of stuff in there, so maybe just only have a look at the first 4 columns there. So you can see that we have 92 rows. ACC is pretty small in terms of patient size here. So that's why I like to use it as the example. So you have 92 patients, and here are some of the columns that are in the call data. All right. So let's have a look at the questions and see what we have. Question: why do we have sample metadata stored in call data and in sample map? Couldn't all sample information be stored in a single object? Well, the single object is multi-assay experiment, and we wanted to have a separate sample map so that everything can be tracked through the sample map and make it easier to subset. Call data will give you the actual days to death values, but the sample map includes only those row names in that call data and the call names in all of the experiments, along with their assay names or assays. So, so sample map sort of keeps that that information sort of isolated, and it makes it easier to, with all the subsequent operations to subset and coordinate the data that way. Good question. Next question from Sonali. Have you considered making pre-existing MEA objects for TCGA available? I'm not familiar with MEA objects. Sorry. So no, but if you can ask offline and we can look at that and see what that's what's required to get those available. Multi-experiment assay. I'm not sure what that is actually. I'd have to look into it. I don't have a good answer. Sorry. Question: who will and how frequently will the cBioPortal data be packaged? So the data is hosted on AWS, and as far as I know, they're constantly working on it. So any improvements that they make, they repackage the data that's hosted on AWS. So might be a better question for the cBioPortal data team. Not too familiar with their workflow, but they make a lot of changes and updates to the data. So I imagine that it's frequently updated and packaged. Okay, so multi-assay experiment. If I understand your question correctly, we already provide these through Experiment Hub. So that is the main way of publishing our data. Annotation Hub, we don't have, I guess we don't have Annotation Hub resources. So we mainly provide our data through Experiment Hub. Sorry if I'm not understanding the question correctly. Now let's take a poll here and see how many people have used some of this software already. So let's look at. Are you familiar with curated TCGA data? Okay, so it looks like it's neck and neck at half and half almost. No is taking the lead. I can show on stage, I guess, here. Now no is running away with it. So yeah, that's totally okay to not be familiar with curated TCGA data, and sorry for the typo. But looks like we should know some of the functionality provided there. Okay, So let's go back into our studio session. So we're working with this ACC data set here, and maybe to introduce some of the functionality that we have in TCGAUtils. I'll add that here so you can load the package. And we'll do something like sample tables on our ACC data set. So you can see that this gives you sort of a crude representation of the samples that are in each assay. So it's good to see what's in there because you may want to remove those and only get the tumor samples, or restrict to only those, because here we have some normals included as well. So we do have a function for that, and we can always do help package equals. Have a look at all of the functions in the package. So we can do TCGA primary tumors, which is a convenience function, so only select the tumors out of a particular curated TCGAData resource. So when you run that, it will find all the tumor samples and restrict to those and remove everything else. So that's what this does here. And the way we can verify is if we run our sample tables again on that output. So that. Now you'll see that everything is 0, 1 here in our output. So that's some of the functionality that we have. You can also use split assays if you want to keep those normals in your data, and that will shift all of your so it will add another normals assay into your multi-assay experiment if you want to split by those tumors and normals. Okay, so that's some of the functionality in TCGAUtils. I will go back to our main vignette here. You can press back if you had that up, or you can go and do help package equals multi-assay workshop, and that will give you the page here. So we're looking at the main vignette. It's integrating multi-omics data with multi-assay experiment, and we'll go through some of the examples here. So we looked at matched assay experiment, and maybe we can try that on our ACC data set if we don't have any replicates, but let's see. So we add ACC matched assay experiment, and then run that. So because we have replicates in our data, it tells us to resolve those. It could be just that we have those normals in there. So I'll create this one using the primary tumors only, and then use ACC2 and see if that's difference. So it does. So if I only select the primary tumors and then do matched assay experiment, and that works fine. So you can see that now we have 45 columns across all the data sets. So we're really limited to only the the assays. So here our PPA array has 46 normals, or 46 samples that are tumors. Sorry, and so there would be only about 46 patients who will be recorded across all of these. So you can see that that's the lowest common denominator. So that's why we get about 45 columns in our matched assay experiment. So now we can do things like correlations and things like that. So that's the other thing that we can do to visualize, to see what samples are in our data, is to use our upset samples function on the ACC multi-assay experiment, and that will give us a nice graphic of all of our assays in the multi-assay experiment. So you can see that about 46 patients here had a measurement in all of the experiments, and about 33 were missing that RPPA array, and only 13 had a single copy number alteration information there. So it gives you a breakdown of what's in your data in a nice visual way. So that's upset samples, which is using the upset ARP package. Okay, so we can use matched assay experiment on after we remove our normals. So that makes it a little bit easier to work with if you have a specific workflow in mind. We'll skip this building from scratch demo. Here we did that already. And here we present a multi-assay experiment API cheat sheet. So in order to get an idea of an overview of all of the functionality that we provide in multi-assay experiment, like the way you would subset, what double bracket subsetting does for a multi-assay experiment. It extracts a particular assay from the multi-assay experiment. So if you wanted to only work with one of these, you could, and you can do the double bracket 1 to get that RaggedExperiment object. You could extract a single assay and populate the call data if you wanted to. So if you had a summarized experiment here, like for RNA-seq2 gene here, you could extract it along with the associated call data and work with that as well. And there are other functions here to combine rows and columns in the data. So we did, with match assay experiment, we combined or intersected all of the columns across the experiment. You could do that as well for the rows if you're interested in a set of genes across all of the experiments. And then we have some reshaping functionality to mainly for graphing and analysis purposes. You can reshape to a long format or to a wide format, depending on the need. And you could also combine multi-assay experiment objects into one. So if you have two different ones and you want to put them in one, you could do that with the C function. Does multi-assay only work with TCGA data through cBioPortal? say I have a batch of tissue samples, I have RNA-seq, and yes, yeah, it doesn't only work with cBioPortal data or curated TCGA data. You can create your own multi-assay experiment, as we saw in our tutorial. So you would have to provide the experiment list and a call data and a sample map to coordinate the row names in the call data and the column names in all of the experiments. Another question, how do you know what the 0, 1, 10, and 11 mean normal? That's a good question, and we can look at our data sets in the package here. So TCGAUtils has a couple or a few reference data sets. So if you run data package equals TCGAUtils, that will list what data sets are in the package. We are interested in sample types. So we'll do this and do sample types. Sample. And then print it. So this is our map that will give us a reference of what the zeros and ones means. So the 0, 1 is a primary solid tumor, the 10 blood-derived normal, 11 is solid tissue normal. So that's our reference data set taken from the TCGA website. And they have short letter codes. Great question. Yes. Yeah. Great. Yeah, we provide curated TCGA data that we were able to figure that out. Okay. Good questions. Let's continue with our workflow here. So we looked at the main components of multi-assay experiment. There also is a metadata slot that you can add information. So if you do metadata mini ACC, I think I really like this highlighting and doing control enter on this, but you could copy and paste into your console. And if we do that, you'll get the metadata for our example data set. So PMID, source URL, extra stuff in there. Okay, so we went over curated TCGA data, how to query some data sets from there. And cBioPortal gives you access to more data. You can do get studies, cBio, to have a look at all of the available studies. And so with some metadata there. And we have this argument here, build report equals true, which will tell you whether our functions are working on the data and whether you can build using the cBio data pack or the cBioPortal data function. So API build and pack build will give you that information. And it will also ask you, so if it's not building, it would say, are you sure you want to proceed, even though this data set's not building as multi-assay experiment? And so you'll know that some studies are not supported as multi-assay experiments until we can figure out some of the issues with the data and code. examples, some utilities. So you can convert with TCGAUtils. We have a number of conversion functions like microRNA to ranges. So we'll consult the TXDB packages and convert microRNA to genomic ranges, or gene symbols to ranges, or do a Q-reduced TCGA to take the copy number data or mutation data and provide a range summarized experiment. So you can convert from RaggedExperiment to range summarized experiment with Q-reduced TCGA, and it will do everything. It will coordinate the reference here in the background for you, so that you don't have to. So, and we provide some examples for doing that here. And in some cases, you may have to change the genome annotation. So we looked at sample tables to see what sample types are in each assay. And this only works with TCGA data. So because these sample values are unique to TCGA. So something to note there. And we have an example for splitting your assays. So if you wanted only tumors and normals, you could split, and this probably has to be updated. This should be TCGA split assays, but you can get separate assays for those sample types. And we have an effort to curate some of the molecular subtypes for some of the disease codes in multi-assay in curated TCGAData. So the way you would pull out the subtype map, say for example ACC, which is one of the data sets that we curated, you can see that we have the annotation in the data, in the call data, and a description of what that column in the call data is annotating. So if we check the call data, say for example histology column, you can see the type there. So this curation is not available for all of them, only for a few, maybe about 15 or so cancer types. And we also have functionality for working with the TCGA identifiers. So that's there. Feel free to ask questions. And I think I wanted to go through our subsetting examples. So this is some of the data management that you'll be doing if you work with several experiments at a time. You can subset by genes or by a condition. So if you're looking for only stage 4 pathologic stage values in mini ACC, you can do that, and that will consult the call data and only restrict to those conditions. You can also select a single assay using the single bracket operation and the name of the assay. So I mentioned earlier, double bracket subsetting will extract the object. So you can use numeric index or the more robust name index here. So I'll skip over these and maybe show you the analysis are marked down here. So let me go back maybe. So we have this multi-assay experiment underscore analysis vignette. So you can have some idea of what you can do with multi-assay experiment. So we can look at our upset samples, and we can do a Kaplan-Meier plot based on the days to death data and vital status. So we restrict some observations here and fit the model using SERVBIT and plot the Kaplan-Meier curve here. So I know we're running up on time, so I'll give you, so you can have a look at those vignettes, and you can ask any questions if you have on Slack or on the platform. So I think everyone has the link to the workshop page. If not, it's posted here at the top. Okay, so I think that's all I have for you. I'll open it up for questions, and thank you everyone for coming, and I hope you got a better idea of what we do at the Waldron lab and at Bioconductor.
Thanks, Marcel.
Thanks.
Does anybody have any remaining questions? I think they've been posting them the whole time and you've been answering them, so I don't think that there's probably not anything left, but just to give people a chance. In case anybody's sitting on a question they've been holding on to.
Yeah, the questions have been great throughout, so
Yeah.
thanks.
Moderator should pull us out any minute now. We could both hide.
I can publish or poll everyone so that we have some
What is a poll?
How likely are you to use cBioPortal for your own workflow? The other question. So the question is, there's a discrepancy in the reference genome that was used for these data sets. GDC uses HG38, cBioPortal HG19. So do we plan to pull data from GDC? There are a couple of ways you can do that. You can lift over, and we provide an example in our main curated TCGAData vignette on how to do that. You can also use the GDC Genomic Data Commons package, although it might require a bit more time to get used to. You can do that as well. We don't have any plans to port the GDC data to curated TCGA data at the moment.
Hi, Marcel. I just wanted to let you know I cut out of Seyoon's workshop to come here and help answer any questions if you want me to. I'm here.
Thanks, Levi.
Marcel's been doing great, and he's answered questions throughout. And
yeah, I knew he would.
and other folks were moderating as well, and they usually got to the questions before I could even see them. So
awesome.
Yeah. The only awkward thing is this pause at the end while we're waiting for the tech person to close the close the session. Oh, here's a question.
Question is, TCGA BioLinks pulls data from HG38 as summarized experiments.
That's a good question.
Yeah, we don't necessarily need a function to convert. I think you can just pop those summarized experiments into a multi-assay experiment, and they should work out of the box. You might have to do some sample map munging there to coordinate. That's a great question. If we were in person, you'd get some of our stickers for multi-assay experiment. And, but you can see our sticker design on our GitHub page if you're interested. Yeah, maybe next year. Thanks everyone for coming, and I appreciate your time, and hopefully I'll see you on Slack.
Great job, Marcel.